Melaseq assay, which we are developing as a multi-tissue test to detect melanoma in its earliest and most curable stage. So in my talk description, I mentioned that melanoma is sometimes referred to as Australia's national cancer. It's also New Zealand's national cancer. And just as a brief little bit of history as to why that's so, I have this image here, which is a map of the Earth and the ultraviolet radiation levels. And I think this figure is actually just for one particular day, but you can generally see that UV rays are higher around this middle band of the planet. And when you contrast that against skin pigmentation of indigenous populations around the planet, you can see that there is quite a similarity between skin pigmentation and ultraviolet radiation, which implies that skin pigmentation is somewhat of an evolutionary adaptation. And then when you factor in that indigenous Australians have been on this land for between 45 and I think up to 60,000 years, and European Australians have only been here for around 200, you can see that we haven't had quite as much time to adapt to this source of radiation. Therefore, we experience some of the highest rates of skin cancer in the world. So as I mentioned, we recently just took back that mantle of highest incidence in the world from New Zealand. 2 out of 3 of us will have a skin cancer of some type during our lifetime. Most of them are not going to be melanoma, fortunately. But for a population of only 23 million, there are 1 million, just got to hide this control panel. There are 1 million skin cancer related procedures per year. All this comes at a cost of $1 billion to Medicare, which is our central paying healthcare system. And that's actually the highest cost to the government of any cancer type, which I find sort of extraordinary for a cancer that can essentially be cured with a scalpel and an outpatient procedure if it's diagnosed early enough. And for putting this in people terms, so like last year, 4 people each day died from melanoma, which is more than the road toll. So enormous problem here. So looking at another way to look at, I guess, the size of the problem is to compare it to other cancers. And an important metric is looking at the mortality rate, so the number of deaths per 100,000 people in a population. So this is data from the Australian Institute of Health and Welfare, and it's age-standardised death rates. So it's adjusting for differences in the population, the age distribution in the Australian population over time. And what you can see here is 4 of the top 5 cancers since government records began in 1968. And what I've done here is just adjust everything back to the death rate in 1968 and look to see how trends are changing. I just have to got control panel over my slides. Okay, but okay. So what you can see, lung cancer, prostate, breast, and colorectal. And if you compare the mortality rate of those 4 cancers to the mortality rate of melanoma, you can see there's a really stark difference. And in 2017, which is the last date of statistics I was able to access, you're now twice as likely to die from melanoma as you were when records began. And if you compare it, if you just look at, I guess, the mid-90s onwards, you see a really strong downward trend in sort of lung, prostate, breast, and colorectal cancer, but the death rate of melanoma is still increasing. And this is despite the fact that there has been really a lot of advances in evolution in the way that melanoma, or skin cancer in general, is detected and diagnosed. We had initially, it was just characterized by describing the gross features. Then there were systems set in place to more accurately describe how skin lesions look and behave. In the 90s, there was a focus on the subsurface features, so what's happening under the layer that you can see with the naked eye. In the 2000s, we moved to sort of digital features, and now there's a real push towards artificial intelligence. And all of these approaches have led to longer survival, not necessarily, obviously not a reduction in the mortality rate. But one thing that they have in common is that they're all looking at the outside of the patient. So that sort of, I'll come back to that in a minute. So the challenge, one of the challenges that we see and that we've heard from physicians and patients is not just early diagnosis, but accurate diagnosis. So So here are 3 different skin lesions, and I will say I'm not a dermatologist or a physician, but the challenge is everybody has a different baseline skin tone, different types of complexion, and that really does affect how different skin lesions will look to a dermatologist or a GP. So here are 3 different skin lesions. One of these will not kill you, one of these will kill 1 in 1,000 people, and one of these will kill 1 in 10 people. And the challenge is telling them apart and telling them apart at the right time. So benign nevi, which is a mole or just a spot, we all have them. Extremely common, never fatal. The other main type and the most common type is the basal or squamous cell skin cancer. These are, like, as I said, extremely common. They are rarely fatal, less than 1 in 1,000 deaths. And then the bad guys are the melanomas, which sort of on the flip side are rare but often fatal. So the challenge is identifying the melanomas without cutting out every single spot or lesion on an individual. So that we've sort of got, with crunching all those numbers, we thought, okay, well, where does the variation come into the system? So we've looked at how melanoma is diagnosed, which is, as I mentioned, like a purely visual approach. So the current way is either the person or the person's partner or a GP or a dermatologist will identify a suspicious lesion. If they think it needs to be excised, they will excise it or send it to a clinic to be excised, and then that goes to a pathologist. So when you look at the variation in that process, as in what is the percentage of lesions that will receive a different clinical interpretation by a GP or dermatologist, you can imagine it's quite difficult to study, but for the studies that we've looked at, there's up to 40% variation in lesions, in the interpretation of skin lesion in the community by GPs and dermatologists. When that is excised and sent to the pathology lab, which is sort of the gold standard method of diagnosing a melanoma, what we see there is there's up to 48% diagnostic imprecision in the pathology process. So that figure comes from a really interesting study that was performed in 2017. I'm just going to run through a couple of stats from that. So this study was inspired by the researcher's own personal experience as a patient. She had a lesion cut out and sent it off. It was sent off for pathology analysis. Being a scientist, she asked to have it triple reviewed, and from that lesion, she got 3 different results that ranged from benign through to invasive melanoma. So decided to set up a study where she took 284 skin biopsy cases, enrolled 187 practicing pathologists, and as a nice add-on to the study, she sent the same cases to the same pathologists 8 months apart. So that's over 106,000 data points. This was the most extensive study of the full histopathologic spectrum of melanoma, involving pathologists from all across the country and in a range of clinical settings. It was also the first study to look at intra-observer reproducibility. So those are where she sent the same cases back to the same doctors without telling them they were the same cases. And then she also compared the individual pathologist results to the results of a 3 pathologist reference panel, a reference diagnosis, if you like. And one thing that I think is important, the results were adjusted for years of pathologist experience. So it was a really well designed study, and I would encourage you to look at it if you're in this field or don't know about it. What she found is that in this set of over 100,000 skin biopsy diagnosis, 45% of cases had a different diagnostic interpretation between any pair of 2 pathologists. It was done in a